Imagine a world where viruses aren't just destructive invaders, but potential allies in the realm of genetics. In 2025, scientists at Virginia Tech made an astonishing discovery while examining a seemingly ordinary green alga, Chlamydomonas reinhardtii. Nestled within the alga's genetic code, they found a colossal dormant virus, now named Punuivirus. Unlike typical viruses that attack and destroy their hosts, this one has an extraordinary ability. It can integrate into the host's DNA, remain inactive for extended periods, and occasionally reactivate without causing harm. This unique behavior has far-reaching implications for our understanding of viruses, evolution, and biotechnology. But here's where it gets controversial. Could this discovery revolutionize gene editing technologies, potentially surpassing the capabilities of today's well-known CRISPR systems? Let's dive in. The discovery of Punuivirus. The journey to uncovering Punuivirus began when researchers employed long-read DNA sequencing on Chlamydomonas reinhardtii, a model organism frequently used in laboratories to study photosynthesis, genetics, and cell biology. Within the alga's chromosome 15, they discovered a massive stretch of DNA measuring about 617,000 base pairs. This genetic segment turned out to be the genome of a virus, making it one of the largest viruses ever identified. To verify the virus's activity, scientists used electron microscopy. To their surprise, they observed fully formed viral particles emerging from some algal cells, even though the cultures appeared healthy. This indicated that the virus could switch between a latent state, where it remains inactive within the host genome, and an active state, where it produces viral particles. Unlike typical lytic viruses that destroy their host cells upon activation, Punuivirus seems to coexist peacefully with its host, reactivating only intermittently and without causing massive cell death. Unique characteristics. Several features make Punuivirus truly fascinating. 1. Gigantic genome. At 617 kilobases, it is far larger than most viruses. This allows it to carry a wide variety of genes, making it a treasure trove of genetic information. 2. Integrase enzyme. The virus encodes an enzyme that enables it to insert its DNA into the host genome and later remove it again, a rare feature in giant viruses. This enzyme could be a game changer in genetic engineering, allowing for precise and controlled gene insertion. 3. Fanzor nucleases. These are specialized proteins capable of cutting DNA, guided by small RNAs, much like CRISPR enzymes. They provide a built-in mechanism for precise DNA editing, potentially offering new strategies for gene manipulation. 4. Latent lifestyle. The virus does not behave destructively. Instead, it lies dormant most of the time, which allows the host to thrive while still carrying the viral DNA. This peaceful coexistence is a stark contrast to the aggressive behavior of many other viruses. Significance for gene editing. The discovery of Punuivirus may prove revolutionary for genetic engineering. Precision integration. The virus's integrase could be adapted as a tool to insert large genetic fragments into host genomes more accurately than current technologies allow. This could open new avenues for genetic modification, making it easier to introduce complex traits into organisms. Beyond CRISPR. While CRISPR-Cas systems have transformed molecular biology, they have limitations, including off-target effects and restrictions on which DNA sequences they can target. The RNA-guided Fanzor nucleases in Punuivirus may provide new editing strategies with different specificities, expanding the toolkit for scientists. And this is the part most people miss. The potential for these nucleases to offer more precise and versatile gene editing could be a game-changer in the field. Gentle delivery system. Because the virus coexists with its host without destroying it, researchers may one day engineer it as a safe viral vector for delivering genes into algae, plants, or even other organisms. This could revolutionize gene therapy and agricultural biotechnology, making it possible to introduce beneficial traits without harming the host. On-demand activation. Its ability to remain silent and then reactivate suggests it could be used as a controllable platform for gene editing. Switching editing functions on only when needed. This could be particularly useful in scenarios where precise timing of gene expression is crucial. Ecological and evolutionary implications. The discovery of a dormant giant virus also has wider biological significance. Viruses like Punuivirus may act as agents of horizontal gene transfer, moving DNA across species and potentially accelerating evolutionary changes. Since algae play a central role in ecosystems by producing oxygen, recycling nutrients, and forming the base of aquatic food webs, the influence of such viruses could shape not only genetic evolution, but also global ecological processes. Field studies have shown that similar viral DNA exists in algae from lakes in Sweden and coastal waters in the Netherlands, proving that Punuivirus is not just a laboratory curiosity, but part of natural ecosystems. This means viruses of this type could be widespread, silently influencing the genetics of algae worldwide. But what if these viruses are more than just silent passengers? Could they be actively shaping the genetic landscape of our planet? Future research directions identifying triggers. Scientists aim to determine what environmental conditions, such as light, nutrient levels, or stress, cause the virus to switch between latent and active phases. Understanding these triggers could help in developing controlled gene editing systems. Functional studies. Researchers are mapping viral transcripts and proteins to better understand which genes are responsible for integration, reactivation, and host interaction. This knowledge could pave the way for new biotechnological applications. Biotechnological development. Work is underway to see whether the viral enzymes, especially the fans or nucleases, can be adapted into practical gene editing tools. These enzymes could offer new possibilities for genetic manipulation, potentially surpassing the capabilities of current technologies. Biodiversity surveys. Broader ecological surveys may uncover more dormant giant viruses in other algae or protists, further expanding our knowledge of viral diversity. This could lead to the discovery of even more unique viral characteristics and potential applications. The discovery of Punuivirus, a giant dormant virus hidden inside green algae, represents a major step forward in both virology and biotechnology. With its ability to integrate smoothly into host genomes, remain dormant, and carry specialized DNA-cutting enzymes, it challenges traditional ideas about how viruses behave. Most importantly, it offers new hope for gene editing technologies, providing alternatives and complements to CRISPR. As research continues, this silent viral passenger inside algae may one day become a powerful tool in medicine, agriculture, and synthetic biology, shaping the future of genetic engineering and our understanding of life itself. But what do you think? Could Punuivirus be the key to unlocking new frontiers in genetic engineering? Or are there ethical considerations we should be aware of? Share your thoughts in the comments below.